My name is George Garnett, and I'm a master's student at the University of Edinburgh and Royal Botanic Gardens, Edinburgh. Um, so in 2016, it was a great honour to win the NBN Gilbert Youth Award for terrestrial and freshwater recording. And here's a picture of me looking very proud and younger at the NBN conference in Edinburgh that year. So I've been asked to speak a little about what I've been doing since winning. So I'll do that, as well as talking a little bit about the flora of Guernsey, where I grew up, because everyone likes plants. So in 2017, I started my undergraduate degree in ecology at the University of Reading. And there I was lucky enough to be involved in undergraduate research in collaboration with the RHS, focusing on the phylogenetics of the Psiloidae subfamily of the Asparagaceae, which is the asparagus family. So I'm sure you all heard of squills, grape hyacinths, and bluebells. These are all members of the Psiloidae, alongside a whole host of other, perhaps less familiar taxa. And I could talk about this group all day, but I've only got 5 minutes, so I'll just sum it up really quickly. Many of the taxa in this group have traditionally been separated based on floral morphology. However, previous molecular work by some authors called Vosser and Spater shown floral morphology to be a rather poor indicator of their evolutionary relationships. So you might think the common garden squirrel seen here would be closely related to our native autumn squirrel, but in actual fact, it was shown to be more closely related to our native bluebell, which I think is really interesting. So the aim of our work was to see if the conclusions of these past molecular studies held up when more sequence data are used. It's early days still, but preliminary findings seem to suggest that traditional concepts of Scilla based on floral morphology are not natural groupings. So in other words, Scilla is polyphyletic. This is a work that got me hooked on plant systematics. I'm really fascinated by how molecular evidence can turn seemingly sensible morphological classifications on their head. And it was a really great opportunity to learn skills such as DNA extraction, PCR, and analysis of Sanger sequencing data. And I'm very grateful to the University of Reading, Europe scheme, and the RHS and Dr. Alistair Collum for allowing me to get involved in this. So now we have the technology to sequence entire genomes relatively quickly and relatively cheaply, which is something that's only become possible in my lifetime. I think that fits in well with today's theme of changing times. So using this next generation sequencing technology, we assembled the whole plastid genome of the bluebell, Synthoides non-scripta, all 155,000 or so base pairs of it. And this was published. So if this is something that interests you, I definitely encourage you to check out our paper. I use this same whole genome sequencing technology for my dissertation research on the genus Muscari. Again, like Scilla, using floral morphology, Muscari has traditionally been split into 2 to 4 genera or subgenera, depending on who the author is. In any case, I tested molecular support for these morphological groupings. Here's 4 commonly accepted morphological subgenera. And in short, molecular plastid data showed that whilst morphology is a good indicator of evolutionary relationships, it's not perfect. Caveat here is that further work's being conducted at Reading, and conclusions may change on that. I think they're also starting to use ribosomal data now, so that should be quite interesting to see. But despite my research leaning towards the molecular side of things, I still consider myself a field botanist. Due to COVID restrictions, I went home to Guernsey in March and stayed put pretty much all summer, which was a great chance to see some of the island's rare plants, many of which I've not seen for years. So a beautiful orchid here on the right-hand side is Anacanthus laxiflora. It's reasonably widespread worldwide, but in the British Isles it's only found in the Channel Islands. and this photo really doesn't do justice to it. Whole fields turn purple in spring. It's really beautiful, and this is in no small part due to the stellar conservation work of the Société Guernessaise, who manage their fields with volunteers and a herd of Guernsey cows, purchased specially for conservation grazing. Ferns were also a group that I became really interested in, mainly because of remarkable fern hybrids found in Guernsey, one of which is Splenium microdon, a hybrid of lanceolate spleenwort and hart's tongue fern. And it's completely bizarre looking. If you know what the parents look like, it's very much intermediate. But I wonder how many people walk past it.
1 minute, George.
Okay.
Please.
So we've also got Isoetes hystrix, which is admittedly not a very charismatic species, but it's really special. It's the only corewort that's not fully aquatic, and it's exceptionally rare in the British Isles. Unfortunately, it looks like several other plants that grow in exactly the same habitat, including autumn squill, and it's tiny, so it's easy to see how a species like this could disappear without anyone knowing, which of course is why we need skilled biological recorders. Following on from this, my final plant that I'd like to show is the Guernsey century, Zaculum bacillum, a species not found in mainland Britain. I saw it for the first time this summer, mainly because it has a tendency to disappear for years at a time, and then reappear when conditions are right. I don't think it's been spotted in about a decade, though like Isoetes, it's tiny and possibly overlooked. So the wonderful Vice-County recorder for Guernsey happened to spot it this year, which was just as well because a wildlife pond was about to be created over the site by well-meaning conservationists, which would of course have destroyed one of the only British populations of this species in the space of a few months, which I think is a sobering reminder not just of the importance of biological recording, but also making these records available to all stakeholders. So, I'll end as I began, back in Edinburgh, and still as keen a botanist as ever. Thank you.
Great.
Hi everyone, I'm Gina and I'm a master's student at both the University of Portsmouth and University Centre Sparsholt. My master's research project involves finding and counting jacksnipe using a handheld thermal imaging camera and identifying their habitat preferences. So today I'm going to be giving a bit of a background and overview of my project. I was asked to talk about my use of existing data and the NBN Atlas, so I'm going to talk about how I've used existing jacksnipe records in my project so far. And then I'm going to give a little evaluation of what I found to be the benefits and limitations of these datasets for me and my project. So the current population estimate for jacksnipe wintering in the UK is 110,000, assuming that one individual is present per kilometre squared over 110,000 kilometre squared of suitable habitat. But this assumption is based on no scientific data at all. The common standards monitoring method for population assessment of jacksnipe are winter transect counts in suitable habitat. But jacksnipe numbers are probably underestimated in these transects because they're so difficult to find and see. So you almost have to be standing on a jacksnipe before it will flush. They make little to no noise. They are extremely well camouflaged, as you can tell in this photo. They're active at night and really good at hiding in the day, and their habitats are wet and boggy and often inaccessible. So this is my working title, which hasn't changed since I wrote my proposal about 4 months ago, when I was young, naive and extremely ambitious, thinking I might actually be able to make a national population estimate during a 12-month master's project. But what I do want to establish in this project is whether thermal imaging can reduce jacksnipe detection difficulties, and whether it can be used as a method or to supplement existing methods to achieve a more reliable population estimate for jacksnipe in the future. And then later on in the project, I'll be developing a habitat holding index or habitat suitability index. So the broad stages of my project are to identify and arrange access to sites, and then carry out habitat and jacksnipe surveys and develop the index. So I'm just at the end of the first stage, identifying and arranging access to sites. I had to find sites that are known to be good for jacksnipe to make sure I actually find some and to make sure I don't just waste my time. So to do this, I made a data request to HBIC and also made use of the records on the NBN Atlas to identify where jacksnipe have previously been recorded. To increase my chances of being able to find jacksnipe, I prioritised sites with the most numerous and most recent records. Okay, so this is my evaluation and pros and cons of the data in relation to my project. So HBIC had 2,118 jacksnipe records and the NBN Atlas had 892 records in Hampshire. A limitation was that grid references were imprecise at 4 or 6 figures, making it difficult to work out where the jacksnipe might have been in those squares. Some HBIC records included notes to help narrow down the exact location, which was good, and I liked that the NBN Atlas had info about the source of the data, so you could potentially contact the source for more information about particular records if you wanted to. The sites with the most numerous and recent records are most likely biased towards popular birdwatching sites and towards daytime sightings, which is great for me because I'm focusing my data collection on daytime roost sites. I read a paper where the authors used the NBN Atlas to look at changes in reptile abundance and distribution over time, but they found that the recent increase in observer effort caused their results to be misleading. But this increase was perfect for what I needed it for, to find numerous and recent records. The NBN Atlas had the most recent records up to 2019, whereas the most recent HBIC record was in 2018. It would have been good if I'd have been able to add layers to the NBN Atlas map, like priority habitats layer, to cross-reference sites with habitat info, and then the time to access. So there's a standard 2-week processing time for HBIC data, but they did get the data to me sooner than that, which is amazing. But the NBN Atlas data is instantly available, so that's even more amazing. So in conclusion, I now have 15 sites secured with between 1 and 4 survey areas in each, a few of which you've seen dotted around the presentation. The main limitation for me was the grid references. If they were more precise, site identification would have been quicker, and I might have even been able to use the records in my analyses. But an increase in observer effort is really great and was really helpful for what I needed it for. My next step is to survey habitats and jacksnipe. And thank you for listening.
Okay, so I'm going to talk to you today about some research I've done for my PhD thesis about range-shifting species. And just if I could ask everybody not to share this on Twitter, because the results are currently being submitted to a journal. So hopefully you'll be able to read about them very soon in that, and it will be completely open access. So if you're interested in the topic, please consider following me on Twitter if you're interested in a link to the paper. So what is a range-shifting species? So sure, everybody knows at the moment that species and the areas they live aren't constant. In fact, over time, there's a lot of movement. And the principal driver of this is climate change, but also there's other factors like habitat change and changes in nitrogen and other differences, environmental differences driven by human and natural processes. And this has a lot of implications for us as humans. We can think of stuff like carbon sequestration. We can think of, obviously, the more sort of relational values we get from nature, the sort of joy of seeing species around us and indeed the health benefits that can bring us. So in this work, I wanted to find out a little bit about what people thought about these species moving into new areas, and specifically the UK. So illustrated with this picture of a recorder doing some acoustic surveying, I held up a microphone to try and understand what UK wildlife controllers were thinking. So what did we find? We found that recorders generally had quite positive attitudes towards the arrivals establishing in the UK, and they varied with different taxa. So perhaps unsurprisingly, we found that birds were amongst the most popular, whereas there was some more concern about bees and wasps and the true bugs, mostly around sort of the potential for pests. Though we did also see in the sort of text answers we asked people to give, that there was also a lot of understanding about the ecosystem services that those species provided in terms of pollination and for wasp pest control. So I think this is really exciting, and one of the reasons is that these new species are a really important opportunity to reach out to new people in conservation. So I'll hopefully show you this quote. For me, experiencing the little egret colonization was very exciting, mostly because they were exotic Mediterranean birds and they arrived by themselves. No huge sums of money thrown at them. None of this rubbish as per white storks at Knepp or ospreys at Rutland. Sorry, obviously, I didn't write this. I think there's some great stuff happening at Knepp. This was the real deal. They colonized by themselves, marvelous additions, and not detrimental to other wildlife, apart from small fish, obviously. And I think that really captures sort of when natural processes are happening, it is something slightly different to when there's human intervention. And there's also this quote about the hemiptera, which is sort of maybe represents some of the reason why there's a difference in attitudes. I've not heard of this species and know nothing about it. So we also asked them about potential perspectives on how range shifters should be managed. And we presented 5 options, sort of from not intervening to adapting and trying to help them arrive, to trying to prevent their arrival and possibly their impacts on native species. And here we found that there was quite a strong preference towards non-intervention and just observing what was happening as the range shift developed. There was slightly more support for trying to support range shifters than there was for trying to prevent them, but it was still quite minor. And some of the reasons, strategies to stop them might be akin to stopping a tsunami with sandcastles. There's quite a lot of skepticism about our ability to manage range shift to begin with, that it's very complex, and then obviously that people are more inclined to spend resources trying to protect threatened species, which obviously makes sense. And then again, this attitude that it's not something that humans should be intervening with, why mess around with them, would be a complete waste of money attempting to hurry up what is inevitable anyway, lol. So in conclusion, we found that range shifting species, it's a broad thumbs up, but management of range shifting species was met with slightly more skepticism. I'd just like to thank my supervisors, Dr. Regan Early, Dr. Nick Isaac, as well as Dr. Sarah Crowley and Dr. Rachel Turner, who helped me design this survey. Thanks very much.